So the last time that we spoke, last month, we did an assessment of hormones, saliva hormones, and today we're going to be looking at mucosal barrier assessment, specifically the intestinal mucosal barrier, and we're going to be looking at intestinal permeability, inflammation, dysbiotic behavior, food sensitivities, etc. And the following month, just to let you know, we are going to finish this series with a look at a metabolic wellness profile. We're going to be looking at a number of features that you will find interesting. But today is mucosal barrier, and I should say that all of these tests are blood spot tests. So a little bit of blood can be taken from the finger and can be put onto a card, which you get in the kit. And from that, you let it dry, and then you send in that card to the lab. So let's take a look at mucosal barrier assessment, also known as intestinal permeability. In order to situate permeability in the larger scheme, we need to look at a slide which I use quite often, and it's the pathogenesis of both inflammatory problems and autoimmune disease, which of course is an outgrowth of chronic inflammatory situations. And as with everything, there is always a genetic predisposition that has to be looked at. Genetics always plays a role, in some cases more, in other cases less, but it is always there. The other thing that you need to keep in mind are the various environmental triggers, which could be anything from food proteins to the infections which an individual may have, the antibiotics they're taking, etc. After that, you need to be aware of the microbiome, especially when it's dysbiotic, when you're beginning to get a dysfunctional microbiome, and specifically the area of Candida, which we're going to discuss in a few moments, and how that leads to immune response, sometimes a dysregulated immune response, meaning an immune response which is not the normal type of response that you should have. And all of that leads to intestinal inflammation in and of itself, which is the localized or regional phenomenon in the gut. And that in turn will lead to, in many cases, an increase in intestinal permeability, and with that, the loss of a normal integrity of the barrier. When you have that, you then are very much primed for generalized inflammatory disease, and with it, even autoimmune problems, which of course also come from the intestinal inflammation itself. And then it becomes a push-pull situation, almost a vicious cycle, in which more inflammation causes even greater amounts of intestinal permeability, etc. And so what we're going to be looking at today, of course, briefly, because we only have about an hour, is we're going to be looking at the biomarkers for intestinal permeability, in this case, zonulin, which many of you have heard of, and also histamine and diamine oxidase, its breakdown enzyme. We're going to be looking very briefly at food sensitivities as well as Candida, and look at that particular fungal entity within the microbiome. The intestine, as all of you know, is the largest surface of the body by far. It's about 300 to 400 square meter area. It's about the size of 2 tennis courts. And when you take a look at that, what you're seeing is something that almost looks like a barrier reef, where you're seeing all of these tentacles moving out into the lumen of the gut. And that is where the microbiome lives. The microbiome has well over 150 trillion, and that's not a mistake in printing. It's trillion bacteria, which is at least 15 or even 20 times the total number of human cells that we have in the body. It outnumbers them massively. And that's just the bacteria. If you start looking at the protozoa or the fungi or for that matter, the virome, which are the viruses, they are 100 to 1,000 times that of the bacteria itself. So suffice it to say that there are more of these little critters in the gut than there are stars in the universe. So it's a massive area, continuously exposed to the environment. So you're getting both bacterial and various dietary antigens that are constantly being looked at by the body to decide if they are friend or foe and what they need to do with them. And because the intestine is constantly having food and other items going down the lumen, it also has an immune system which is in intimate contact with the barrier itself. In fact, 70%, and some people even say 80 or 90% of the lymphoid tissue in the body is in the area of the intestine. And given its important function, 40%, fully 40% or even more of the body's energy expenditure is in the gut itself for obvious reasons. A healthy gut environment is one in which the lumen has both commensal bacteria with a certain amount of pathogenic bacteria, pathobionts, which are under control within a healthy environment. They live in the mucus layer of the gut. It's actually a bilayer, an inner and an outer layer. And there you have this wonderful connection and network of bacteria, of these trillions of bacteria that are communicating amongst themselves as well as with the rest of the body. When you have an unhealthy gut environment, either due to antibiotics, the kind of diet that you have, the hygienic conditions of both the environment, the food that you're eating, etc., the various pollutants, the viruses, you can end up with a dysbiosis, which means that you're getting not just a decrease in the commensal bacteria and an increase in the pathogenic bacteria, but really a much more complex interrelationship, which is beginning to cause sufficient damage to the epithelial barrier that it is allowing for the entry of macromolecules, either bacterial entities or various dietary macromolecules that should not be getting in that form. And we'll discuss that in a few moments. So let's take a look at the intestinal IQ tests, as we call them. And these are made up of 4 tests. And we're going to first look at the Zonulin test, which is the test that we look at to judge intestinal permeability. Now, we know that when you look at intestinal absorption, the mucosa of the intestine, that one cell lining, one layer of cells, it's remarkable that it is only one layer of cells, and it has a very paradoxical role. On the one hand, it is trying to absorb nutrients, but at the same time, it needs to be able to exclude various toxins and macromolecules so that the body is only seeing the particular molecules that it requires for its nutrient function. And so if you get a breach of the mucosa, for whatever the reason, you end up with any number of things, including malabsorption of food entities and, of course, a much greater increase in exposure to both toxins and the various antigens. So what is the real definition then of intestinal permeability? Well, we know that when you have movement across the gut wall, there are really 2 pathways that are well known. The one on the bottom here is called the transcellular pathway. That is the classic transport of micromolecules, of the various small entities, either food or whatever the body wants to take in, and that comes in through the cell itself. But between the cells, you have a paracellular area, or intercellular, whichever way you've learned it, and there you have diffusion of various types of peptide entities, etc. Now, these peptides getting in are controlled by the opening of this paracellular pathway, and there happens to be an area which we call the tight junctions. It's a network of proteins which act like a door that can be opened and closed. It is a defense mechanism that the body developed for a very important reason, that if something is sitting on the other side of these cells that the body wants to get rid of, it needs to be able to flush those microorganisms or small peptides that it doesn't want. It needs to be able to flush them out. And so the way it does is it puts them into solution and flushes them out. And so that door opens. But if the door is open to let things out, it can also let things in. And so you do have these tight junctions, and there is a particular proteinaceous entity in the body, which is called zonulin. And zonulin, which is found in all mammalian species, is in fact the molecule that controls the opening and closing of those tight junctions. It is the only physiological modulator to date of this intercellular tight junction area that's been described so far. And what it does is it helps control macromolecule movement, because I'll reiterate, the body does not want to take in macromolecules. It wants to break them down into micromolecules. And when macromolecules do get in, it needs to be able to get them back out. And so it is ultimately one of the modulators of tolerance to immune response. The idea being you want to be able to have a situation of hemostasis, of tolerance, and you do that by having an immune system that is able to function only when it is absolutely required in order to be able to get rid of those entities that one is not looking for. So let's take a closer look at this paracellular tight junction area. You see it here on the right. It's made up of a whole series of proteins together in a network, actually a net itself. And it was once considered that it was very static, but we now know it's far from that. In fact, it's just the opposite. It's quite dynamic. It can open, it can close, it can adapt to any number of problems, developmental problems, be they physiological, pathological, any number of dietary circumstances. All of that will open and close this tight junction area. As I said earlier, it's a defense against various toxins, various infectious agents, which if they do get in, you want to get them out very quickly. You also want to occasionally absorb certain nutrients passively. It can do that as well. It is always surveilling the gut population in the lumen of the gut, and at times will want to bring in certain complexes of antigens, that is, bacterial antigens that have been complexed with antibodies like IgA, and they will be brought in so that they can be dealt with by the innate immune system. And it's also there to transport white blood cells into the gut when you're dealing with an inflammatory situation. So if you're looking at what affects intestinal permeability, and by the way, I'm going to use this word once, it's been known affectionately as leaky gut. We don't like to use that term normally, but many of you may remember that. That was the non-medical term for that. So we're looking at augmented or increased intestinal permeability, and it can be caused by any number of these things which you see on the left of the slide, but especially on the right: food allergies, malnutrition, dysbiotic behavior. All of that, together with these others, will lead to elevated total amounts of toxin and antigen burden that the body needs to deal with. And if it cannot deal with it properly, you enter into not just a local inflammatory response, but something that enters into systemic disease or chronic inflammatory problems. And there are many situations, diseases that are associated with zonulin, the various autoimmune diseases, which we now are beginning to understand much better, the various inflammatory bowel diseases, of which the poster child are the two diseases, Crohn's and ulcerative colitis, celiac disease, diabetes, type 1 and type 2, by the way, rheumatoid arthritis, lupus, ankylosing spondylitis. In the nervous system, multiple sclerosis very clearly has a problem with intestinal permeability. And I'm going to mention autism. We can discuss that later, but I can tell you that there's more and more evidence that one of the features of the pathogenesis of autism is intestinal permeability problems and massive dysbiotic behavior. So there are a whole number of conditions which you will see in your PDF that are part of what used to be called leaky gut syndrome, increased intestinal permeability. And we're not going to discuss it in great detail today, but if you wanted to have a longer lecture, we go through in detail all of these particular entities, both inflammatory problems and frank disease, including celiac disease, IBD, etc. And let's take a look at celiac disease specifically as an example of what I talked about before with what is the pathogenesis of these problems. In the case of celiac disease, there is a genetic predisposition. 95% of celiac disease patients have HLA-DQ2 or 8 receptors. They have more of those receptors on the presenting cells in the immune system so that they are primed to be able to respond much more robustly to any entity which they would want to respond to, in this case, to alpha-gliadin, which is part of gluten, and gluten is part of wheat. And so that is the classic environmental trigger, which is the second part of that. That in turn causes a very robust microbiota response. And in fact, the microbiome becomes dysbiotic with all of the attendant problems. That causes an immune response which is outside the norms of what you would normally expect. And that will create increased intestinal permeability, in some cases very, very dramatic increases in intestinal permeability, which creates not just the intestinal problems, but the problems that celiac sufferers have throughout their body. It is a generalized problem of the whole body, but it starts in the gut. And what is causing it? Well, we say gliadin in the diet, which, as I mentioned, is a component of gluten. But in fact, the reason that happens is that the body is perceiving gliadin as a pathogenic organism. In a sense, it doesn't see it any differently than it would a pathogenic bacterium. It's just responding in that way. And so from that comes the dysbiotic behavior, which is the same that it's doing with any number of the different parts of the microbiome, be it the bacteria, the fungi, the parasitic entities, whatever that happens to be. So if you're looking at the factors that are driving the immense rise, we have an epidemic, folks, of inflammatory and autoimmune diseases that are occurring right now. What are they? And I'm just giving you a slide which is going to encompass all of that. We spend a lot of time and lectures specifically going through all of this, but you can put it into the following summary. If in the first 2 to 3 years of an individual's life, which are critical, by the way, if you have had a vaginal delivery, if there's proper nutrition, if there has been very low amount of infection, and with that, very low amounts of antibiotic therapy, definitely less than 2 times per year in the first 2 to 3 years, then the microbiome composition is going to be quite balanced. That first 2 or 3 years is critical. Whereas if the person was delivered by C-section, improper nutrition, especially not getting the mother's milk, multiple infections which called for multiple antibiotic therapy at least 2 times a year, then you are going to almost certainly have dysbiotic behavior. If you have a balanced microbiome, then there is a very high threshold to the immune system in the lymphoid tissue of the gut, which is what you want. You want there to be a high threshold because you don't want the immune system to be shooting off at any time. You want it only to come in when there's a very serious problem. But when you have an imbalance, well, with a high threshold and with a genetic predisposition, of course, there's therefore a normal response to antigens. You have stable health. You don't have a problem. However, if it's an inappropriate response of the gut-associated lymphoid tissue, that means it has a low threshold. You're going to end up with immune responses at a very low threshold. That is not what you want, and that is going to promote pro-inflammatory responses to all of the antigens coming in, which of course will lead inevitably to chronic disease and various types of epidemics. And so we've discussed zonulin, which, as I said, it's a protein found in intestinal cells, and for that matter, it's found all over the place, in the brain, in the heart, in the skin. Almost all cells have zonulin, and I will tell you it is one of the first line screening tests that one needs to do for gut status, that is for sure. The second one that we're going to look at is histamine and diamine oxidase, and this is important, and it's one of the reasons we offer it as an important part of our testing profile, is because it is a very good and quick way of being able to get a handle on intestinal inflammation and the immune response to that. We've all heard of histamine. It's a very well-known, one of the best-known biogenic amines, and it's involved in any number of things. Local immune responses. It regulates the physiological functioning in the gut as well as other tissues. And of course, we also know it's a very powerful neurotransmitter. And histamine is synthesized from L-histidine. And histidine is what you find in many, many foods. You also find it in many bacteria, and it's also synthesized by histidine in the mast cells, which are one of the major components of the innate immune system, right at the gut level, and of course many other cells, basophils, the platelets, and the enterochromatophin cells, which are some of the specialized cells of the mucosal barrier of the intestine. And so it is stored in little vesicles and little bubbles inside these cells, waiting to be released if there's a need. And when histamine is released, there needs to be a way to get rid of it, and the way that you do is through 2 pathways. Histamine can be metabolized by oxidative deamination, which is the classic way, and that is diamine oxidase. And diamine oxidase is found in large amounts in the intestine, and we're going to discuss that more in just a few moments. The other way is through methylation, and that is histamine N-methyltransferase. We're not going to discuss that today. We're going to focus on diamine oxidase. Now, diamine oxidase is a very interesting molecule. It is a protein in the cell plasma membrane which scavenges extracellular histamine, specifically the histamine that is found extracellularly in the lumen of the intestine. So diamine oxidase, which we're calling DAO activity is quite unique among intestinal mucosal enzymes. And the reason it is that the circulating levels of DAO are a marker, it's a biomarker of mucosal maturation and integrity. If you want to know the integrity of the barrier, this is one of the biomarkers. And in fact, serum DAO activity correlates very well with intestinal permeability of the small intestine, which is a critical component in digestion. And in fact, if you are having a problem with the intestinal damage, initially there's increased DAO activity, but eventually if that continues, you begin to get decreased DAO activity because the cell itself, its physical architecture breaks down. And so that's why both histamine and DAO and the ratio of the two is extremely important. I'm not going to go over the overview of the small intestine. What I am going to tell you is that the cells that make DAO are found in the crypts. Those are the valleys right at the bottom of these little villi. These are these microscopic hypha or little fingers that move out into the lumen of the intestine. And they are normally very beautiful long fingers, unlike what you see in inflammatory bowel disease and IBD, where you don't have them. It's completely flattened out, which is one of the big problems in IBD, that it doesn't have the capacity to be able to properly control the kind of thing that DAO would normally control. So normally, and this is on the bottom, in a physiological condition, DAO is produced by cells in the crypts. They proliferate, and cells are always moving from the crypt up to the tip of the mountain of cells. And so as they're maturing, they're proliferating, and you're getting more and more DAO. At the same time, it is able to tell these cells what is going on with the various types of nutrients that are coming in, especially the short chain fatty acids. So as you are getting increases in DAO, what it is able to do quite well is to decrease dietary histamine. That's its main job, to destroy excess histamine so that you don't end up with the problems of too much histamine. But when you have a problem in a pathological condition, which I'm not going to discuss in detail here, and there is not enough DAO being produced, that allows histamine to increase. And so bottom line, when you're taking a look at what goes on with DAO, what you need to understand, and this is an important slide because it says intestinal barrier damages can occur due to a whole number of problems. They can be mechanical. Literally, you are mechanically destroying these cells. Microbially, which is one of the main ways it happens, especially with pathogenic bacteria. Enzymatically, especially when you don't have enough mucus covering the cells. And that damage can occur due to genetics, of course, due to dysbiotic behavior, due to infections, especially if they're ongoing, the loss of the immunoglobulin SIG-A, that's secretory IgA, which needs a functioning mucus layer in order to function well. If that's not there, then you've got a problem. You've got a decrease in the various types of proteins that are able to metabolize these various bacteria. And of course, an increase in histamine can cause the problem, and it's coming due, as I said, to any number of reasons: the mast cells, microbial, the diet itself, stress. And if that happens, and if you do get enough damage, you get a loss of barrier integrity. And with that loss of barrier integrity, you will start getting a lot more mucosal inflammation, as well as an increase in intestinal permeability. As soon as you've got that, you have a vicious cycle now that continues. That is the initial problem. And so with early mucosal injury, you would expect an increase in diamine oxidase. Why? Because there is damage to the mucosal cells, and so they begin to create a flood of these particular entities as part of the inflammatory reaction. But if it goes on long enough, you begin to get an increase in apoptosis. That means death of a cell, especially the crypt cells, those cells which are producing DAO. And when that happens, no more DAO is being produced, or of course it goes down initially. And so it's down both in the lumen, and you also see it down in the blood. And so that is a late mucosal injury, a decrease in DAO because of the destroyed or dysfunctional mucosals. And so we do know then DAO is synthesized, as I said, in the epithelial cells of the villi and the crypts, and damaged mucosal cells will release DAO, which initially will increase its serum concentration. And you need to be aware that that happens in the beginning, and then after that, you get a decrease. And so what I'd like to do is now take a look at Candida. Now, Candida, we know, is a fungus that has its home in the gut. And I think it's very important to recognize that Candida in and of itself is not a problem. Candida is part of a large commensal community in the gut. And normally it is found in large amounts in the body, and it is part of the normal microflora until it's not. Until you get sufficient damage to the microbiome, which allows certain, in effect, holes to develop in the total amount of the microflora, and that's going to be taken up. It fills the space, so to speak, that was left by commensal bacteria beginning to either disappear or to not function properly. And so it begins to colonize the mucous membrane in much greater amounts. It then can pass into the host's bloodstream, and that stimulates an immune response and, of course, an antibody production against Candida. And the way in which one looks for that is both through blood tests and through stool tests. The blood test is by far the best way to look for it because it checks for the three main antibodies to Candida, IgA, IgG, and IgM. And it is most often the preliminary test that you would want to do because it's able to reflect an activated immune response. And is it acute? Is it a chronic infection? It'll give you that information. The stool test actually checks for the antigen, the Candida antigen, and it is most often a part of a comprehensive stool analysis. And you will see it when there is a significant antigenic level of Candida. What you do need to remember is that there are three immunoglobulins that respond. IgM, which is the first one, it's the first immunoglobulin that is going to respond, and it activates another pathway, which is called the complement pathway, which will produce a very robust immune response. Then there's IgG, which we're all familiar with. That comes on later. It lags by a few weeks or even a few months in certain cases. It is what we know as the memory immunoglobulin. It also activates complement. And of course, IgA, specifically secretory IgA, in the lumen of the gut. And its primary function is within the mucosal tissue of the gut. That's where it lives. Normally, it does not escape from there normally and will not normally show up in the blood. unless there is a very active infection. And here is just another way of saying it. IgM, as a screening test, you've got this Candida suite, the 3 antibodies, and IgM you'll see increasing at the beginning of an infection. IgG develops later, so it indicates a past infection or a prolonged infection. And IgA, as I said, because it's almost exclusively in the mucous membranes of the gut, when you do find it in blood, that means there is a significant ongoing infection which has really created enough of the IgA that it has spilled over into the blood. We are presently in our lab developing an advanced Candida suite, which is also going to test for the spherical versus the hyphal forms of Candida. And if somebody wants to ask me that question later on, I'll be very happy to answer why we're doing that. Very quickly, we're not going to discuss food sensitivities, but our lab does offer both 24 and 88 total IgG food sensitivities for those who do want to use it. And the reason that we offer it, frankly, is because it gives us a handle, gives the healthcare professional a handle on enzymatic imbalances. Looking at the various groupings of foods, you're able to tell whether there are enzymatic problems that are emanating from the stomach. Is it coming from the pancreas? Is it a bile issue? Or is it a combination of all three? And you can find that when you learn how to read these food sensitivity profiles. So if you're looking at a complementary assessment, a primary screening, and let me reiterate, these are screening tests. These are not diagnostic tests. None of these are, but they're extremely important. Next month, we're going to be discussing this first one, which is called the Metabolic Wellness Profile, and it is a dried urine test, and it tests for digestion, for liver detoxification, and cell oxidative stress. I'm not going to say any more except that that is an extremely important test, and I give all of these tests that I'm discussing, I give them what's called a usefulness rating of 5-star, meaning that really you should be looking at these as extremely important primary screening. The barrier assessment, what we just discussed today, the permeability changes, the damages to the tight junctions done with dried blood spot. This is the blood zonulin. Again, a usefulness rating of 5. The inflammation and permeability change, which we just discussed today, histamine, tells you if you have inflammation in the gut. Why is it there? The diamine oxidase, is it breaking down histamine well enough? Is it showing you that there is a permeability change? And if yes, where are you at that point in the damage to the cell? This is extremely important, by the way, in determining what avenue of treatment you're going to choose. There's a very big difference in what you do with late intestinal barrier damage and how that differs from early barrier damage. Again, 5-star. Activation of the immune response, as I mentioned, food sensitivity, we test for total IgG, and it is identifying, if you remember, the complex of the antigen and antibody. That's what you're looking for. And as well, the Candida, as I mentioned, the 3 immunoglobulins to that. And last but not least, what we did discuss last month, and I believe very strongly should be part of your testing armamentarium, and that is to test for the stress response, the salivary hormone profiles, a critical feature in being able to determine how cortisol and DHEA are affecting the issues. In the case of intestine, I think you all are aware that all 3 of the sex hormones have an effect on intestinal function, intestinal healing, and so it is very important to get a salivary hormone profile. This is something that we have in the back end of our website, and just to show you what our test look like? And if you do get a test, you're going to see on the first page, depending on what you order, the zonulin, you could get the DAO and histamine, the ratio of the DAO and histamine. If you've ordered the food sensitivities, you would see that separately on another page. And you take a look, we give you the various elements here. Here you see zonulin, again, DAO, histamine. We tell you what the result is in the box. Where does it sit in the horizontal bar, which tells you where it is within the reference range. And that gives you a very quick idea of where that individual is. And so when you are looking at, for example, zonulin, we know that if you're in the green area, and I'm just giving you a general look at this, if it's somewhere between 1 and 6 nanograms per mil, that'll tell you that you have an inflammatory problem, maybe. You may have some intestinal drying. You're losing some of the mucus, which means that you'll have a deficiency in Sig-A. That's not a small point. It's important, but it's not sufficient to cause an intestinal permeability problem. That's very different from what happens as you enter into a borderline area where you're now getting much more definitive gut inflammation. Often that's due to dysbiosis, various types of food and enzyme imbalances, and it's now beginning to affect the permeability. You would like to be able to catch your patients or clients at this point because it's much easier to treat them at this time. What you don't want is to enter into a period where they are already in an elevated area, somewhere between 10 or 20 or even greater, where you have all the signs and symptoms of permeability of the tight junctions due to any number of dysbiotic, enzymatic problems, hormonal problems, etc. It is quite established at that point, and that is telling you that you must deal with it at that point. It can actually go way above range. We have people who are undiagnosed celiacs or diabetic conditions where they are sitting way above 20. They can be in the 30s and 40s. These are people who have acute problems that must be dealt with. Same thing with histamine and DAO. When the histamine levels are low, that is optimal. The inflammatory problems are insufficient to cause, due to histamine, any kind of intestinal permeability. So there is some inflammation, but it is quite localized. That is quite different from when you get very high levels of histamine, where you're getting increased gastric acid secretion or even permeability changes. Quite often it's due to food that's been ingested, as I said earlier, that is very high in histidine, which is going to create histamine. Many bacteria will have the histidine, and that'll create histamine, especially, for example, seafood, crustaceans. Seafood like fish that's been left after the fish is dead, you will get histamine increases. It'll double every 20 minutes. So that's why if you have a dead fish, it has to be gutted, meaning the insides need to be taken out very quickly. And that's extremely important in plants that deal with fish, etc. So that's extremely important. Just as we talked about histamine, DAO is like the mirror image. When the levels are very high, because here you see that high levels are in the green, you want higher levels because the higher levels are needed to be able to break down elevated levels of histamine. And normally, DAO will not increase to higher levels unless there is higher histamine. That's what it's supposed to do when it gets higher. But if it's very, very low levels, you then have to say to yourself, why is that happening? Is it because there is a problem of intestinal permeability and it's broken down the architecture of the cell? What is it that's causing that problem? And so in the interpretation, you will see a a DAO to histamine ratio, which many of your colleagues use, because that's going to give you a very quickly an idea of whether or not there's a problem. Is it an optimal ratio? For example, is it such that you don't have a problem? That can be even if DAO is becoming slightly problematic, but when there's very low histamine, it's still okay. That's very different from a borderline area where the DAO may be higher, but the histamine is very high. So now you look at both of those and you say, wait a second, the ratio is getting very close to red. And then you have the various types of very high ratios, where it can be due to very high histamine, even in the face of very low DAO, or what's even worse, extremely high DAO with extremely high histamine. That means you've got the worst of worlds. You have histaminic intolerance together with increased permeability. That must be dealt with very, very quickly. I did mention to you the food sensitivities. I'm not going to go over that again. And I told you about Candida. And this is what you would see in our report. You would see IgM, IgG, and IgA. For example, in this case, IgG is non-reactive. It's less than 9. There's very little or no significant antibody level of Candida albicans. So that means whatever is going on, it has not been a long-standing infection. It's not prolonged. It may be something that has just started recently. IgM, as I said, shows you there's a presence of antibody. They're not sure. It's still indeterminate because it's between 9 and 11. If it was greater than 11, it would tell you there's probably a Candida infection happening at that moment. Let's say IgA is picked up. Here you have 14.2. That's much greater than 11. So there's Candida albicans that has been detected in the form of secretory IgA. So it is indicating either a past or a very active or prolonged infection. Clearly, that needs to be looked at. We also provide with our company, for those who are with us, a large manual of about 40 or 45 pages, which goes through all of the different protocols for treatment, which you are going to find very interesting. And in fact, we have in the back a spreadsheet which includes biotics research as well as many others, so that you are able to tell which particular type of supplement would you want to use if you're using supplements to be able to deal with the various staging of treatment for intestinal problems. And in the manual, it goes through the 8 stages of intestinal problems and treatment. And this spreadsheet will tell you when will the supplements of the various companies, when will they act best. And here with biotics, you can see all of these supplements and where it is felt that they would have their best effect. Just as an example, betaine plus HP, here it has its best effect when you're trying to balance enzymes in the stomach, etc. And that is not something we decide. That is given to us as information by the supplement companies. So let me state just at the end that the results in this report are, again, not diagnostic, and they should not be viewed as treatment recommendations. Of course, those decisions are the sole responsibility of the healthcare professional. And with that, I'm going to say thank you.